Good morning, everyone, or good afternoon, depending on where you are. Today, we're going to have an in-depth analysis of the Cystatin C protein structure. Here in A, you can see the primary structure of the protein Cystatin C that's being represented. This is no different than Figure 1, which is represented above. It's just a different schematic to show you and outline the same structure. So as you're aware by now, the cystatin C protein consists of 5 beta sheets, represented here by the 5 arrows, 1, 2, 3, 4, 5, as well as 2 alpha helixes, represented by the cylinders. In addition to this, the L highlighted in pink here represents the leucine at position 68, which is ultimately altered to glutamine in the mutated version, which has significant consequences, as we'll discuss later. In B, we just take the primary structure and see how it folds into a secondary structure. Again, you can see the protein has a longer alpha helix and a shorter alpha helix, as well as the 5 beta sheets that are all represented here. Again, this is the monomer form of the protein. Leucine at position 68 is once again highlighted in pink. One thing you'll notice here is that the leucine is at an integral position within the monomer, meaning it's at the very center of the protein. Something we'll discuss later on is that the center of the protein is very hydrophobic in nature, and leucine, when it's altered to glutamine, which is a hydrophilic acid, amino acid by contrast, it creates a repulsion within the center of the protein that really causes it to unfold and alters its function. Moving on, we see here the tertiary structure of the protein. So the monomeric protein undergoes partial unfolding and dimerization via domain swapping to ultimately adopt the dimeric form depicted in figure 2.2. So this is just an annotated version of the diagram depicted in figure 2.2 to better help understand the positions of all the hairpin loops and beta sheets and alpha helices. Cystatin C consists of several conserved domains. The substrate binding sites of cystatin C are extremely conserved and located in beta hairpin loops that join strands 2 and 3. So here you can appreciate strand 2 and 3 being joined by this beta hairpin loop here. This correlates to site 1, as well as strands 4 and 5, which join via this loop here to create site 2. Ultimately, this dimeric form of the protein has several functional properties within the body, including biochemical and biological, as well as neuroprotective properties. When this structure is altered due to the mutation of the leucine at position 68, better represented here by the red dot, as well as here, the structure is ultimately unfolded. This unfolded structure then, in a way, kind of oligomerizes with other proteins to form deposits within cerebral arteries. Again, we'll discuss this later on. It's just a foresight into some details that will be coming. If you have any more questions, please feel free to look through our website regarding the structure of cystatin C in particular, as well as hereditary cerebral hemorrhage, which results from this specific mutation at position 68 with the conversion of leucine to glutamine. Again, thanks for your time. If you have any questions, I'll provide an email in the website. Shoot through your questions there. Thanks.